Hi, everybody, and thank you for inviting me to participate in ISME Latin America. I really wish I could be there in person, and I also wish I could be giving this talk to you live, but I can't, and so I'm recording it about 8 hours ahead of time, so it's almost live. Because I can't be there to answer the Q&A for you, I would like to introduce Taichi Suzuki, who is a senior postdoc in the lab, and a lot of his work I will be showing you. He did the work on co-phylogeny, which is towards the second half of the talk. Taichi is a postdoc with us since 2018. He did his PhD at UC Berkeley, and he did a lot of work during this time on what's shown here, the microbiome of mice, and how the microbiome of mice has helped their adaptation to new environments, and how they affect their phenotypes and so on within a species. And so he has he brings a fascinating view of evolution, and he's transferred that to human evolution. And so a lot of the ideas that I'll talk about in the second half of the talk in particular are really coming from Taichi. So he's very much a thought leader in this area, and keep an eye out for him. You can find out more about Taichi from his LinkedIn page. He writes also a blog, and he will be there to answer questions. So I hope you'll have a fun time interacting with him after you're done watching this talk. So my group works on the human gut microbiome. And very quickly, because I think you already probably know all this by now, we emerge as beings, relatively or completely germ-free, and we're rapidly colonized by the people and maybe the pets around us. We are also introduced to microbes that come from the environment, from the food we eat, but the ones that really stick, that really become the human gut microbiome, look to be very much acquired from people and unique to people, if not our close relatives. So what this means as you grow up is that you have an increasing complexity and diversity and biomass that builds up, so that by the time you're an adult, you have a very high number of microbial cells and in the gut. And we talk about mostly the large intestine when I talk about the microbiome in my group. So you'll have this mix that can be largely unique to you, although of course shared with other people as well. And there's been a lot of work over the years, in the last 10, 15 years, about what actually can contribute to what particular people might have. So that ranges from what people eat, to their lifestyle, to their levels of health, to the communities they come from. And in my group, we've been focusing on the role of host genetics and genetic variation between people, and how that might impact not only the diversity there and the structure of the communities, but more recently even getting into very specific molecular interactions. So we started off asking this basic question: does host genotype variation matter at all in shaping your microbiome? And another way of phrasing this is, do people have a particular genetic predisposition for the microbiome configuration that they might have? And coming into this, so this was work started in my lab around 2008 when I started at Cornell. The prevailing answer at the time was no. Host genotype really has no influence that's appreciable. And this came from comparisons at the time out of Jeffrey Gordon's lab of twins, monozygotic and dizygotic twins. This is showing unifrac metric, so an overall idea of how similar different diversity can be for pairs of twins. And these data indicated that really host genetics was not an important factor in shaping the overall diversity of the microbiome. So this was based on a broad metric of microbiome comparisons. It's also based on what, for today's standards, is a fairly small sample size. And so we decided that perhaps we needed more twins to get a better idea of this. And this was the work of my student, Julia Goodrich, at the time. We paired up with Tim Spector's group at King's College London, and they run a very large twin registry in the United Kingdom. And we were able to sample over 1,000 twin pairs. And these were both fraternal and identical, so dizygotic and monozygotic. And the good thing about working with this group was that these people that we sampled were also genotyped, and our initial work was to profile their microbiomes, and so we ended up generating about over 3,000 by 16S ribosomal RNA sequencing, and this provided the basis to be able to ask if there were microbes that responded to host genotype. And so the analysis runs something like this. Twins are either monozygotic or they're dizygotic. And when they're monozygotic, they share a genome. And so if you measure a trait, this could be height, it could be abundance of Prevotella, and you compare the values you get for the two twins within a twinship, the variation that you might see is going to come from the environment alone, because their genetics are the same. If you take dizygotic twins, the variation that you might see comes from not just the environment, but also the fact that they only share half of their genes on average. And so there are models that you can apply that take this kind of data into consideration. And of course, we're controlling for all kinds of other things that affect the microbiome. But for each and every microbe that we can get an abundance for across the population, we can ask, is the variance in the proportion of the relative abundance of this microbe influenced by host genetics or not? And so this is all summarized in one graph here by Julia. So she's run this ACE model of heritability on the relative abundances for all of the taxa that are represented in this tree. And she's calculated the effect of the host genetics, and that's called the heritability. So it has nothing to do with inheritance. It's a term that's used in genetics to say, can we predict this trait if we know something about your genome, or is that irrelevant? And so that's scored from 0 to 1, and then it's painted on here as a color. So you can see a lot of blue and greens, and this means that most of these branches on this tree, showing mostly bacteria, some archaea, mostly knowing host genotype does not help you predict the variance for the relative abundances of all these taxa. But then you'll see some red branches peeking out. And these are the ones for which if knowing host genotype will actually help you make a better prediction. So that means that people with different genotypes have different abundances of these select microbes. So this is consistent with what was seen before. Overall, if you average all this, you won't see much. But there's important ones that seem to tell a story here that we can look into further, that are picking up something important about host genotype and reflecting that in their abundances. So at the time, we made a list of our top significant heritable taxa. And this is a lot of the list shown here. What I'm showing here is that since then, since that paper in 2014, I'm sorry, and then this is actually getting out of date too, you can see that these taxa are shown again and again in different studies around the world to be heritable. And so the list that we came up with wasn't just something that was unique to English people, but we're seeing the same taxa reported as heritable in different studies now from different parts of the world. And so that is nice validation that there's something perhaps unique and interesting about each one of these. And it gives us a roadmap, if you will, of types of microbes to go after and to ask, what is it about host genotype that these guys are picking up? I'm just going to tell you a little bit about two that we've followed up on in my lab right now as a little segue. So Methanobrevibacter and the family Chrysanthiaceae are organisms that we focused on mostly because this was our most highly heritable, and it co-occurs with this guy. So that's shown here. So the family Chrysanthiaceae was our most highly heritable taxon in this study. And this is a co-occurrence network showing other things it co-occurs with. And so it might not be too surprising that the things it co-occurs with are also heritable. A lot of these, we don't know what they are. They have no culture representatives. But this group here is the methanogens, and we definitely know about methanogens, and Methanobrevibacter smithii is the dominant human methanogen. So what is it doing co-occurring with these? And these are relative newcomers in the sense that there's been a cultured isolate only since 2014, and they've only more recently, if you will, gotten a name. We've been looking for these since to make sure that this co-occurrence wasn't, again, something unique to English people. Alban Rouault in the lab has taken a couple thousand metagenomes from 10 studies and looked again for the relationship, and they do co-occur across all of these various studies, and this co-occurrence is at the family level, the genus level, the species level. There's something with this group, these two groups, that they co-occur. Generally, it looks like, even here in South America. Another thing that, another reason these piqued our interest is because we also looked to see in our original study if there was any relationship to BMI. And this whole cluster is significantly enriched in lean compared to obese individuals, which was a surprise. We didn't expect this. And this led to questions about what might their function be. We've since seen that again in other studies as well. So for instance, Alban, these 2,000 or so metagenomes she was able to look at had information about age and BMI. And both of these are also not only highly prevalent across lots of different people, but they're associated with a low BMI. We've since kept track of various studies that pick this kind of thing up. This is a recent review of which studies report now an association with a low BMI, and you can see that popping up in various parts of the world. If you add healthy metabolic state, you add even more studies and even more parts of the world. So it looks like there's something about these taxa. They co-occur, they're heritable, and they associate with a healthy metabolic state repeatedly. I just want to point out real quick that when you're doing this kind of search, it's important to keep in mind the biases of the databases. So if up until a certain point in the literature, there's no mention of Christensenella because the name didn't exist, then with 16S, it starts popping up in the literature, and you'll find these associations with health markers. And then it disappears again for a while because people switch to metagenomes, and a lot of the reference databases that were being used for this didn't include the genomes. And then it's reappearing again. So just because a name appears or disappears doesn't mean that it wasn't there in the data. It's just the lenses we use to find these things. And to help with this, Nick Youngblood and Jacobo de la Cuesta have developed some tools, and you might find them useful, to build your own custom databases. And so this is something we had to do originally when we were looking for the Christensenellas in metagenomes, for instance. So take it over at our tools. Some of them you might find useful if you do this kind of work and need to build your own databases. Okay. So back to these guys. They're heritable and they're co-occurring. And what can I tell you? Well, Christensenella minuta is the type strain for the Christensenaceae. There's not very many of these in culture at this point. They're relatively recent. There is an expanded, uncultured diversity in this family that looks very interesting, for which there's a lot of mags. But this one is the one we started working with in the lab. And it basically takes sugars and ferments them. Methanobrevibacter smithii is a methanogen that takes hydrogen and CO2 and makes methane. So you can see that there's potential here for cross-feeding. And in fact, when we grow these two together, they love growing together. So C. minuta in culture by itself will form these large biofilms. And M. smithii, it turns out, loves to co-aggregate with it. It doesn't do the same if you put it in with something like Bacteroides theta iota omicron. Now, if you zoom in on something like this, and this is the work of Sofia Elizondo in the lab, you'll find that the Seminuta makes these dense rosette structured biofilms, and then M. smithii, you'll find it right in there contacting those cells. So these bigger ones here are the M. smithii. If you grow them in a bottle, Seminuta will make a lot of hydrogen, and the methanogen, which is fluorescing in orange here, will use that hydrogen. So this is hydrogen increasing over time, decreasing over time, methane taking off over time. They're very easy to grow in co-culture. So Sophia has run these in steady state, growing them with glucose as the sole source of fermentable carbon for the Chrysanthemella. And we can choose to add or not excess hydrogen and CO2. And so then she could compare what Seminuta does alone to what they do together. And this is the kind of thing she sees. So if Seminuta is alone, it grows to this level. These are now genome equivalents per mil. If M. smithii grows alone with CO2 and hydrogen that we provide, it grows about this level. But together, they grow to much higher levels. They use more glucose, they produce more biomass, and interestingly, Seminuta produces less butyrate, which is something that we're currently looking into as to what it is that Seminuta is picking up on to change its metabolic output. And we know at this point it's not hydrogen related, so it's something else. We've also in the past, and we're following up these experiments now, taken Seminuta and added it to a microbiome from an obese donor that lacked it. So we can add it dead or we can add it alive and mix it in and then give that to germ-free animals. And what we find doing this is that the animals that get either heat killed or some kind of control have less weight gain and less total adiposity compared to the animals that don't receive Chrysanthemella added to this obese donor microbiome. We also see some shifts in the metabolic output of that community. So that's some introduction to what these two might be doing, the Chrysanthemella and the Methanobrevibacter. And this is an active area of research in my lab. If you're interested, keep an eye on us for that. What we can ask when we have this list of microbes that we know respond to host genotype is, well, can we identify in the human genome what genes they might be picking up on the variation of? So if you think about genes, there can be differences between individuals when you compare a particular gene. There can be single nucleotide polymorphisms, for instance, that will change the amino acid content of a particular protein, for instance. And so we're looking for SNPs, or these single nucleotide polymorphisms that could lead to actual changes in gene function. And so what we do for this is the genome-wide association analysis, where we take the abundance in each and every microbe that we have from the twin data set, and now we ask, does it relate to the differences that we see at the SNP level in the hosts? So the thing is, there's about 2 million SNPs, and so we're talking about 2 million tests per microbe. So it's a lot of tests, and you have to reach a very high threshold of significance in order to pass the significance bar, because you can imagine there's a lot of false positives that can come up when you're running 2 million tests per microbe. But one, and I should say right now that any genome-wide association analysis done with microbiome as a trait is underpowered at this point. We just don't have the numbers. So from a human GWAS perspective, they do, the geneticists are doing these kinds of analyses on hundreds of thousands of individuals. We are lucky if we have in the thousands. So we do it anyway because we can. And the thing that's been interesting to see is what's popped up from this. And what has popped up from this most strongly again and again and again in every single underpowered GWAS of the microbiome is there's a signal coming out of chromosome 2. So if you're unfamiliar with a Manhattan plot, this is all the 2 million tests that were done for Bifidobacterium the p-values thereof lined up by the position of the SNP on the chromosome. So you've got your human chromosomes lined up. So the SNPs in order across the chromosomes, and these are, you're looking at a couple million p-values. And you're looking for p-values that are really, really, really low. And everybody gets this peak on chromosome 2. If you zoom in on chromosome 2, you'll find that in this region, it's the region that controls lactase persistence. It's the lactase persistence locus. And so your genotype at this locus can predict whether or not, when you're an adult, you are continuing to make lactase, which is the milk sugar for lactose, or whether you shut that down. And so this is strongly correlated with whether or not you're lactose intolerant. So we found this is data from our study. This has been seen again and again in studies of Europeans and North Americans. And I should say, so far, there's only been studies of North Americans and Europeans, and that includes Israelis as well more recently. So in a sense, it's just become sort of a positive control for your GWAS. Are you picking up the bifidobacterium lactase link? And when you think about what's happening here, this is what seems to be happening. So if you are a non-persistent genotype, so you have low expression of lactase, when you drink something like milk that's got lactose in it, you have no or small amounts of lactase in your own intestine. And so that lactose is going to reach the bifidobacteria and it's selecting for them. You see more of them in people with this genotype. If you are lactase persistent, and there's a strong chance then, of course, that you don't have lactose intolerance, your own enzyme in your small intestine is still there, produced in adulthood. This is something that now can access that lactose. There's not much making it, or there's less making it to the microbes where the bifidobacteria are, so there's less of this. So there's weaker selection here for these bifidobacterium. And so because bifidobacteria are famous for the use of lactose, you basically have compensation for your own levels of lactase by the bifidobacteria. So they are compensating for the host genotype here. We've looked more in depth into this genus because Bifidobacterium is a genus. There's many species, there's many strains. So there's some in particular that might be driving this. We did this by looking more carefully into some of these twins from Twins UK and generating metagenomes, and even doing genome capture to enrich for bifido genomes and monosequencing and strain comparisons. And I can tell you that whether or not you're lactase persistent or non-persistent, whether or not you have fewer bifidos or more bifidos, it's the same population structure of the bifidos, and we didn't see any patterns at the strain level. And so we can think of this as the genotype being the tide that rises the boats. If you're the genotype that allows more lactose into the large intestine, you will just have more of these bifidos in general. The whole bifido population is lifted. So lactase persistence is something that developed during the Neolithic, so 10,000 years ago, thereabouts. And it cropped up in different parts of the world independently. It's a beautiful story of convergent evolution, I guess. It's one of the strongest pieces of evidence that there's been selection on humans as they spread around the world and as they adapted different lifestyles. So as people developed animal domestication and started to use animal milk, different kinds of dairy animals, different parts of the world, you see this selection for lactase persistence to be able to make this enzyme in adulthood, which means that adults are drinking milk, which is not something that's normally happening in mammals when you think about it. So the basal state for the human population in general is to not do this, to have lactase non-persistence, but in particular parts of the world, it became advantageous to keep your lactase on in adulthood. And that was a direct result of a dietary change, of a lifestyle change. And there are other examples of human genetic adaptation to novel environments. And this is something that Sarah Tishkoff's group in particular has sort of summarized here. This is from Sarah Tishkoff's group, summarizing some of these genetic adaptations that have happened as people encountered novel environments. So that could be in the case of drinking milk, changing diet. It can also be adaptation to cold, adaptation to high altitude, adaptation to predators that are in certain areas of the world, and so on. And so if we have now seen that the microbes are responding to differences in human genotype that have to do with a diet change, maybe we can use this as another roadmap, if you will, to ask how have microbes adapted with people, or how have they adapted to the changes that the people have had in their own genotypes. So another one we've looked at is starchy food. Starchy food is also something that became more enriched during the Neolithic and the human diet in general, especially for people who obviously developed agriculture. And you begin to break down starch in your mouth with salivary amylase. You also produce pancreatic amylase further down. But the process of starch digestion begins with amylase that cleaves the bond in starch produced in the mouth. And so if you use salivary amylase and then can take up the liberated glucose in the small intestine without microbes getting it, then that is a big energetic win. If you allow starch to proceed down into the intestines where the microbes are, and they get it first, they will ferment it to short chain fatty acids, which is healthy. Some of these are great, like butyrate, but it is an energetic loss. And so you can think, because they're taking an energetic cut here, obviously, you get less energy out of this processing of starch. So it's clear that the host enzyme is providing more calories. And this sets up, in a sense, a competition between the host and the microbes for this substrate. Another thing to note is that the copy number of this gene in the human is directly related to its activity. And the copy number can vary between 2 to 20. So there's been duplication of the amylase gene. And for every extra copy that you have, you make more amylase in your mouth and you have more activity. So you might think that someone who has many, many copies of this and makes much more salivary amylase, that person is not allowing as much starch into the hindgut as a person who has a very low copy number. And this is something that Angela Poole in my lab looked at. And you can read the original paper, came out a couple of years ago, and she looked at people with high copy numbers or low copy numbers of this gene. And as you might expect, it has an effect on the microbiome. And so if you have low levels of the salivary amylase, meaning that you have a low copy number, there is more of this accessible starch that makes it into the gut. If you have a higher amount of salivary amylase in the mouth and you're depleting this non-resistant starch, what reaches the gut will be the resistant starch. And there's some microbes that are particularly suited to the degradation of resistant starch. And these are things like ruminococcus. And we see these amplified where the non-resistant starch is depleted by the host. And so if the host gets the easy stuff, only the hard stuff is left for the microbes. Here it's a mix. And so we see more ruminococcus when the AME1 copy number is higher in these types of hosts. And so we see this as another example of there's the genetic differences between the people. That leads to a real difference in enzymatic levels, and that leads to a real difference in the microbes that respond to the substrates that are usually in play for these enzymes. So those are two that we can talk about, and we're interested in looking for more. And what Taichi did when he was thinking about this is realize that for every single one of these human genetic adaptations to the environment around the world, you can find that there's also associations with the microbiome. So there's enough microbiome studies now that if you look at the human traits associated with these genetic adaptations, you can also find associations with components of the microbiome. And so this leads us to think, well, have the microbiomes been interacting with the host human genotype in the process of adaptation around the world? And I very much encourage you to read this review that summarizes a lot of these thoughts that Taichi has had about how the microbiome might be speeding up or slowing down the process of host adaptation. This doesn't apply just to humans. It applies to any organism with a microbiome that is then moving into new environments. People, of course, we know, came out of Africa about 60 to 100,000 years ago, and then spread across Asia, into Europe, across Asia, and then finally into the Americas. And one of the questions we've had in thinking about human adaptation to novel environments and just human dispersal in general is, did the microbiomes go with the people? And can we see that? Can we see long-term vertical transmission of microbes as people moved around the world? And this is very much inspired by Helicobacter pylori. And you know, I think the Helicobacter pylori story is shown here in this original paper by David Falouche. This is the only known instance so far of the patterns in the microbial strains of this, the patterns in the strains of Helicobacter pylori being consistent with patterns of human migration. And so this is based on analysis of isolates that people took, the H. pylori researchers obtained over the years around the world. And looking at the dominance of the different strains around the world, it's consistent with the idea that they originated from Africa and then spread. And this is also consistent with the fact that Helicobacter pylori is known to be very tightly vertically transmitted within families, because it really doesn't survive for more than a few seconds outside of the human body. And so because it's generated, it's transmitted tightly within familial groups, as people spread, they carried it, and we can now see this in the data. So besides H. pylori, are there others? Can we see any others in the human gut microbiome? And can we do this without having to culture? So what we need, one way of doing this is to test for cophylogeny. And so for each microbe that we're interested in, if we collect from various hosts around the world, and we build a tree, and then we have a tree of the hosts, can we see if those trees match? So schematically, if you have a tree of the host and you have a tree of your bacteria and they match, this would indicate that they've co-diversified. You could get this pattern, of course, if diet, for instance, was driving this, but you wouldn't. You might see that you only get this strain here, and you only get this strain here, and you only get the strain here. But what we're looking for is matching phylogenies, and this way of asking if the trees match. So it's telling you that they share a pattern of their history, and if the patterns match more than you'd expect by chance, then it's not going to be just something like diet setting up these patterns, because then you wouldn't necessarily, there's no reason for the trees to match. So this would be perfect matching. This would be less perfect matching, showing that they frequently exchange, that the bacteria have exchanged between these populations of humans. So we've done this already a little bit looking across the mammals. And this is work by Nick Youngblood, who took who generated fecal metagenomes for over 100 species, and these were all collected in the wild, which makes this this data set particularly interesting. And so he has he has hosts that include birds, amphibians, sorry, not just mammals. And here are the mammals, here are the reptiles. And he's run a number of tests, and this is using 16S data to ask, do the bacterial trees match the host trees or not? And what this is showing is that they tend to match better for mammals than for some of these other groups. So higher on the scale means a better matching of these trees. So there's something already coming out of the mammals, and particularly the herbivores here in this particular data set. Andy Mueller and Howard Ockman published a paper a couple of years ago now where they used another single gene, gyrase B. And this is not omnipresent across bacteria, but it gave them data for Bacteroidetes, it gave them data for bifidobacteria. And they collected from chimps, bonobos, humans, and gorillas. And this is the tree of the hosts. And what you can see, and this is an example for bifidobacteria, is very nice mirroring of this bifidobacteria tree with the hosts. Here, there's been another jump, it looks like, between these two hosts. And so you have gorillas showing up again. And so this is used to support the idea that these bifidobacteria actually co-speciated with the hominids. If you look in human populations, and this is straight out of the Strainfland paper, you can see, and this is now using metagenomes from humans and asking how different are the strains of these different species between different populations. And you can see different strains of here Bifidobacterium longum in the different populations. So this is known. What we'd like to know is, does this reflect only environmental differences between these countries, for instance, or lifestyle such as diet, or can we see if we know the evolutionary history of the people, if we have the host tree, can we see a matching of the tree that we would get from these strains with the host tree? And so to do this, what you actually need is matching host genotypes and gut metagenomes. And it's tough to find these in the public domain. So we have generated a set. We collected from adult women and young children in Germany, in Gabon, and in Vietnam. This, you can see them doing things. This is because we did this as part of a study led by Victor Schmidt and Liam Fitzstevens and Mira Wong to look actually at lactase non-persistence. But we had in the back of our mind that we could also do some other types of studies if we had host genotype and metagenomes as well. So we have metagenomes for the mothers, and we have fecal metagenomes for the mothers and the kids. We also have the data I showed you before from the Twins UK, so we can add another set of women here to match the women from these other countries. So we have three populations, Germany and the UK representing Europe, Gabon representing Africa, and Vietnam representing Asia. We have SNPs from the hosts, and we can build a host tree. We have metagenomes, fecal metagenomes, and we can, so far what we've done is use Strainfland with marker genes to make trees for roughly 50 taxa that we can see in a minimum number of individuals here set at 10. So, sorry, 100. So this is really the brainchild of Taichi, and he's been driving this project to compare the trees. So you can compare at the population level, and then you can also look within populations at much finer structure here. So again, when Taichi takes these data sets and makes the tree of the hosts, we see Africa, red, Asia, blue, and we see orange here representing Europe. So Germany and the UK people end up pretty mixed. And you can see Africa basal here, which is what we expect. And so that's based on about 30,000 SNPs. So now what we can do is take the metagenomes, take strain data, and ask for as many people as possible what strain is there, build a tree of the strain, and compare it to the host tree. So here are some of the microbial trees. Here's a really pretty one, for instance, Collinsella aerophaciens. So each one of these represents a strain that came out of these metagenomes. And this is a phylogeny of those strains. And so you can see, and they're colored now by the continent of origin. So you can see right away that all of these African ones are basal, followed by Asia, followed by Europe. So that almost matches perfectly here. Some of these others are a bit more complex. Provotella copperi is known to have radiated a few times. And so we're not surprised to see these bursts of clades. But you'll see Africa, basal, followed by Asia, Africa basal, followed by Asia, Africa basal, followed by Asia, and some Europe, and some Asia, and some Europe, and so on. So we can look at these. Here's another one. The European ones out here, Africa basal. So some of them more or less complex. I'll show you an example of a couple that are completely mixed up. So here are, for instance, some of the Bacteroidetes. There's absolutely none of these nice clumps. And so what we can do now, besides just squinting at them, is say, okay, statistically, how similar is this tree to this tree? And we're not just looking for 3 groups. We're also going to do this within populations. And this is the kind of thing that you get. So this is Taiichi has now run the statistics using 3 different statistical packages, 3 different approaches to comparing trees. And he's ranked them by significance of cophylogeny, okay? So these are the top 7 taxa that show evidence of cophylogeny using all 3 tests. And what's particularly gratifying is that if we just look within a population, we can see the same thing for some of them, particularly in Africa, somewhat in Asia, less in Europe. So Europe has very low genetic diversity, which might be one reason that we're not picking up the signal so much. But what this does support is the idea that you have long-term vertical transmission, and it's not just these dietary differences maybe between populations, because then you wouldn't see cophylogeny within a population. So we've wondered how general these findings are. And what we've been able to do is use public metagenomes that include both human fecal metagenomes and primate fecal metagenomes to ask if we see some of these same patterns in a slightly different way. But here are the trees now built with public metagenomes. And these are the top 7 taxa built now with public metagenomes. And you can see for some of these, for instance, you see Africa basal, followed by Asia, Africa basal, followed by Asia, Europe out here. So you're seeing that a lot of these patterns are maintained even when we add a huge amount of data to this. And there's some particularly, particularly interesting things that have come out of this, anecdotally, perhaps, such as, for instance, if you look at Madagascar. And so these Madagascar is this bright red on the outer ring. Madagascar has been populated by people from both Africa and from Asia, but it looks to us like their Eubacterium rectali, for instance, and this is also the case for the Provotella copperies, might be of Asian origin. Anyway, there's some things like this that you can see by squinting at the trees. But again, what we'd like to do is try to look at this statistically. Here with the public metagenomes, we don't have the host genotype, but we can ask about transfer events between continents, and do we see it the way we expect or not? So these are the top 7 taxa in now with public metagenome data added. These are the bottom taxa. So you can immediately see that it's way more mixed up. It's way more mixed up. And so we can ask, if we look at the substructure, do we see out of Africa events more often than into Africa events, for instance? So this is shown here with the original 4 populations. If we take the top 7 and the bottom 7, and we ask how many times do they transfer from Africa to everybody else, from Asia to everybody else, or from Europe to everybody else? What you can see is that the top 7 transfer from Africa to everybody else more than the bottom 7. Interestingly, the Europeans, it's the other way around. The bottom 7, the ones that show no evidence of co-phylogeny, they're transferring more. So they might be actually spreaders that we're picking up. When we add the public metagenomes, we can see this pattern again. It's consistent. So we see the top 7 best co-phylogeny scoring showing out of Africa events. Bottom tend to be coming out of Europe. Now, I told you we collected from the babies, and these are now data from bifidobacteria. These are some of our top co-phylogeny scores from the kids. And you can see some of these same patterns as well, definite clustering by population. And we can also ask, is Africa tending to be basal more often than you'd expect? We can also compare these to the mom's genotype data. So if we compare these patterns to the mom's genotype data, so this is only half the genotype data, but it still gives us co-phylogeny scores for many of these. And we also see it within populations as well. So this is within Africa, within Asia, we lose it in Europe. Many of these, again, are bifidobacteria, which are really common in the infant gut microbiome. But we picked up Prevotella copri. And this was, in fact, one of the few that overlapped with the list we got from the moms. And so we're seeing vertical transmission in the adults and also in the kids as well. And so because we have data from the mothers and the kids, and we have these analyses are telling us that these things might be long-term vertical transmitted, we can, of course, now compare mothers to their kids and say, well, are they short-term within this generation vertically transmitted? And this is shown here. So what we've done here, and this is work by Hage and Liam again, is compare strains between mothers and their own kids to strains between mothers and not their own kids. So what is shown here is 2 different ways of comparing strains of these species using a technique developed by Hage that uses synteny, and then this is using markers. This is using in-strain, and we get pretty much the same patterns using these 2 different techniques. So the blue is mother to own child comparisons, and the gray is mother to not own child comparisons. And so we're looking for strains that are more similar when they're from mother to their own infant. And those are highlighted by the asterisks, which I'm sorry, rendered pretty weird, but these are highly significant markers here and here. And what you see on this is that many of the ones that are the most to give us indication that they are vertically transmitted. So we're looking at identical strains. These are Prevotellas. There's definitely something going on with Prevotella. If you work on Prevotella, you've picked an interesting organism. It's giving us these wonderful signals of transmission between mothers and infants, and then transmission over extremely long periods of time. We haven't published any of this yet. And in fact, I've talked about it, Taichi's talked about it, and we got a call from Laure Segurel, working with Rand Blackman, who said, hey, we've got data from Cameroon that looks a lot like your data. You should add it. This was irresistible. So we have now added Cameroon data to our analysis. These are now, we had from Gabon forest dwellers. They also have Cameroonian forest dwellers. These are showing up in the human tree as being related to each other, but as opposed to the other Cameroonian and Gabonese. And so we're currently rerunning everything, and we hope to have something on bioRxiv that you can read if you're interested in the next few months. So what has this given us? It's given us a list of microbial taxa that have evidence of co-diversification, both between and within populations. We see this supported by additional populations and by mother-child strain sharing. And what we think then is that we're picking up on gut microbes that have ridden with their hosts as over the long term as people moved around the world. So this sets up a possibility for reciprocal adaptation and coevolution even between hosts and their microbes. So you can ask Taichi about this. Coevolution is kind of a loaded term in that it's difficult to show that gut microbes meet the standard for that. But if they have been moving with the same hosts over time to such a degree that we can pick it up with this kind of analysis, it does suggest that there has been an opportunity over many generations for coevolution. And so we can also ask what makes these strains different from the others. Do they have special roles, and so on? So this is what we think about. We're trying to link together vertical transmission, thinking about long-term migration patterns, thinking about how people adapted to their environments, thinking about how microbes adapted to the people as they were adapting to their environments. And so we come at this from a few different angles. What I've shown you today was really the broad patterns. We also work at much more detailed mechanistic levels as well. And with that, I'd like to thank the whole extended group. We have a number of people from Latin America in the group. In fact, we have over a dozen countries represented. So we're including Uganda, Australia, you name it. So we're a very international group. If you're interested in working with us, please reach out. We're supported by the Max-Planck Society. And these are some of the collaborators that we've worked with in the past on the twins, with Lars Angenant for bioreactor help. And Sophia worked with Lars Angenant as well. And then here are collaborators that we've worked with to sample around the world. And with that, I'm going to stop this presentation, and thank you very much for listening to it.